Thank you all for coming out here tonight. It's rainy and cold and not very nice, so it's a great time to come and sit in this lovely library and hear about crazy things. I'm going to start with a short clip about some of the things that I do. One of the hats that I wear is as a National Geographic explorer, and this is something that I filmed some time ago wearing that hat.
These are the small pieces, which means that the stuff is washed downstream. See, these pieces are actually still frozen in the permafrost. You can't get them out at all, which means they're going to be really well preserved. Just heard that big splash of water back there. That means another hole's broken through. Here comes the water. We better get out of here.
In my defense, that water is disgusting. So I was in this clip in the Klondike Goldfields. It's near Dawson City in Canada's Yukon. And what happens in Dawson is that there is a lot of permafrost. That's this frozen dirt, soil, kind of really fine glacial silt that the gold miners are trying to wash away to get to the gold-bearing gravels beneath all of that. And as they do that, all of these bones from mammoths and giant bears and cats and bison and horses come washing out. And we like to collect those. They don't really care about those bones. They're trying to get to that gold. But they use these big holding ponds of water, and they spray everything down and then recollect the water and spray it back down. But that dirt is tens of thousands of years of rich organic material that has just been exposed to the very warm summer sun of the Klondike, and it reeks, right? It is disgusting. You do not want to get caught in the onslaught of that stuff coming down over you. You will not get rid of that smell, right? So part of my job is genuinely to run away, right? My job is this. I am a paleogeneticist. If you haven't heard that word before, it's probably because I made it up. It means easy. It means paleo, old, and genetics, DNA. So basically, I study the DNA of dead things, of things that used to be alive. But the motivation for this work is because I care about things that are still alive. And if you've been watching the news or thinking about things recently, you will have noticed that we're kind of having some trouble with things that are still alive. We see that there's changes in precipitation patterns that are causing droughts in some places and floods and major storms in other places and pushing lots of habitats and the species that live in them to the brink of extinction. And the headlines that we see about this are often pretty catastrophic and not particularly fun to read. And instead of thinking about how we are screwed, as the New York Post reported in this particular instant, we might want to think as scientists about how species and populations and biological communities and even entire ecosystems are going to respond to the types of changes that are happening to habitats around the world today. As a paleogeneticist, what I do is I look to the past. And this is a plot that shows average global temperatures. This is a proxy. We look at isotopes in ice cores and ask how cold it was at any point from the past, 100,000 years ago, to the present today. And what you see relative today is this dotted line here. What you see is that it's not stable. There's lots of movement. And in fact, over here, what we see is a really rapid warming event out of the last ice age. In fact, in some of the places where I work in the Yukon, we found that there was a 10 to 12 degrees Celsius increase in temperature over the course of about 40 years, around 10,000 years ago. That is an incredibly rapid period of climate change. And we know now that we can get DNA from plants and animals that live throughout this entire range. And with that DNA, we can ask how these species and communities and ecosystems responded to these past periods of climate change, with the goal of learning things that we can then apply to making informed decisions about how to protect species and populations and ecosystems today. So the field I work in is called ancient DNA. It really doesn't mean anything about the age of the DNA. Instead, as you'll see, it means the state, the quality of that DNA. And the DNA that I work with, because it's old, is kind of crappy, right? Right? It's really broken, it's got damage all over it, but just to reset things, to start from the beginning, DNA is a chemical, a long biological chemical. It's made up of 4 different chemicals here that get the labels A, C, G, and T. These are the nucleotides that are strung together to make the long strands of DNA that are in our genomes. In us, and in most eukaryotic species, DNA strands like this are arranged into chromosomes. This is a karyotype. It shows all of the chromosomes of a human. You can tell that it's a male because you'll see 23 pairs of chromosomes here. This is important. Pairs of chromosomes is important, so remember this. This is going to come in handy. They come in pairs because we get one from mom and one from dad when the egg is fertilized, so we get pairs of everything. And we know this is a male because we see over here they have one gloriously long, very wonderful, large X chromosome from mom, and one tiny little Y that it gets from dad. Right? Biologically male, right? So all organisms have DNA, and all organisms are different in their genome sequences. But because they all are made up of these 4 bases, the building blocks of life, they're all fundamentally the same. And what we can do as scientists is we can line up 2 sequences of DNA and count the numbers of differences between them. And we can use those differences to know how long ago those 2 organisms shared a common ancestor, and even start to understand what it is that makes those organisms look and act different from each other. If I could get DNA from my favorite extinct species, a dodo, for example, and I have gotten DNA from a dodo, and I could get DNA from this other flightless bird, a solitaire, which is closely related to a dodo and also extinct but lived on a different island, and I've also done that. I could line up those sequences of DNA on a computer, and I could count all of the differences between them. And that number would tell me approximately how long ago they shared a common ancestor. And then I could study those specific differences and ask what it is about those DNA sequences that might have led to the different phenotypes, the way these animals look, and how they act. And this is really what we're doing when we're trying to get DNA from ancient organisms. How do we do it? If an organism is still alive, this is pretty easy. In fact, there are companies out there right now where you can send off pay some money, they'll send you a little kit. You spit in a thing and it'll send it off and it'll send you back your genome sequence. It's pretty easy like that with ancient DNA, although the DNA is in slightly worse condition. We know that we can get DNA from lots of preserved elements. We can get DNA from bone. This is a bit of a wolf jawbone that was found by one of the gold miners that we work up in Kansas City. We can get DNA from teeth, from both the calcium, both of the enamel on the outside of the teeth, and also from the roots, which tend to be better preserved. This is a baby mammoth tooth that one of my students found in 2018 when we were working out in Canada. We can get DNA from hair, even hair that doesn't have a root, right? You hear on TV that you can only get DNA from roots of hair. That's because forensics for a long time relied on being able to recover very long strands of DNA, and you can't get long strands of DNA from the shafts of hair, but there is a lot of DNA in the shafts of hair. So if you were thinking of committing crimes, keep that in mind. We can get DNA from any bit of shed hair. We can get DNA from mummified remains of things. This is Zur, a baby wolf that was found in the Yukon several years ago. He's more than 50,000 years old, and we've been able to recover DNA from his hair and his tissues, which we've sampled from the outside. We can even get DNA from processed biological materials. A few years ago, I was working with a librarian who was really interested to try to figure out what animal was used to make the leather binding on a book that was in their collection. And they sent me a little corner of this piece of the book, and we extracted DNA from it using our latest techniques and studied it. And we were kind of disappointed because the only thing that we were able to get out of that book was human DNA. And there's human DNA everywhere, right? And it was, kind of old looking and degraded like you would expect. But, so I wrote to her and I was like, oh, I'm really sorry. We've only been able to get human DNA. I guess this didn't work. And she said, I thought so. Yep, she had sent me a potential human skin book. This isn't that book. This is a different human skin book. This book is actually part of the Wellcome collection in London. So this is a human skin book, not the one that I worked on. The problem with all of those types of DNA is that, unlike DNA from you, this DNA tends to be in bad shape. So if we can think of modern DNA as long party streamers, really nice, lovely, maybe hundreds or tens of millions of bases long at a time, very long fragments of DNA, ancient DNA is more like confetti. And when we're looking at these pieces of DNA, often the only thing that we can recover are fragments that are maybe 30 or 40 or 50 letters long. So much, much more fragmented than the DNA that we get that's long. Why is that? Well, it's because after every organism dies, the DNA in its cells starts to get broken down into smaller and smaller and smaller pieces until eventually there's nothing there that we can recover. This happens because of things like UV light. We know that when we walk outside, UV can actually damage the DNA in our skin cells. That's why we wear sunscreen, so that we don't get cancer every time we go outside. Of course, we have proofreading enzymes that will go along and fix some of the damage that's caused by UV. After you die, there are no more proofreading enzymes doing anything because that's an energy requiring process and there is no more energy after death. And so that damage accumulates. Freezing and thawing will also cause damage to DNA. The physical expansion of water molecules will break the fragments of DNA, causing increasing damage. And most importantly, the actions of microbes, fungi, and bacteria that will get into a decaying organism and chew up that DNA, transforming it into whatever it is that's going to make up the next generations. We know now that there are some environments in which these processes are slow. And this is just like you might imagine in your kitchen. If you get a piece of chicken and you leave it out on the counter, it's going to decay more quickly than if you put it in the fridge. And that's going to decay more quickly than if you put it in the freezer. So a lot of the work that we've done has gone to really cold places in the Arctic where an animal might die and the whole remains, the bones, the teeth, the hair, everything might be buried in that dirt and frozen for tens of thousands or even hundreds of thousands of years. We also know that places like caves are really good for DNA preservation because there's no UV, constant stable temperature. So our job as ancient DNA scientists is to collect this confetti-like bits and pieces of DNA and piece them together into the long party streamers that make up the genomes of the plants and animals that we're interested in, and then use these genomes to better understand how it is that species and populations and ecosystems came to look the way that they do today. So most of the work that's been done in ancient DNA to date has focused on a part of the world in the far north called Beringia. This is because in Beringia, and the place that you saw in the beginning is part of what Beringia was, it was cold, but it wasn't wet enough to be entirely glaciated during the ice ages. And more importantly, this whole bit of land here that's yellow, this is National Geographic yellow in case you're wondering, this whole bit of land here that's yellow, this was exposed because during ice ages, much of the planet's fresh water was taken into making these massive glaciers that sat on top of the continents, which meant the sea level was a lot lower, exposing this bit of land here called Beringia in what's known as the Bering Land Bridge. And this land bridge allowed for the passage of things between Asia and North America. Things like horses and mammoths went from North America into Asia. Things like bison and people went from Asia into North America. And this bit of land was exposed and a refuge for all of the things that we've come to think of as these iconic animals of the Ice Age. Mammoths, mastodons, woolly rhinos, big cats, big bears, all sorts of really cool animals whose remains get deposited in the permafrost in Beringia, and we can go and collect them. We know, of course, that Beringia doesn't look like this today. Instead, it looks more like this. I've actually taken this picture. I'm in that helicopter whose shadow you see on the ground. I'm going to show you that helicopter in a minute because it is top-notch. Really. But my job as a scientist is to figure out why. And not only why, but what can we learn that can help us to make decisions about what to do today? Are there some communities of organisms that are more resilient in the face of large-scale changes to their habitats? What can we learn that we can genuinely apply to conserving ecosystems in the present day? So we fly out into Siberia. I promised this helicopter. Isn't it beautiful? You'll notice that in this helicopter, there's actually some windows missing. Yeah, this was great really good, because after we finally took off in this helicopter, packed on top of these gas tanks that took up most of the inside of these things, the French and Russian expedition team leaders decided to celebrate that we finally got off the ground. It took 3 tries. Finally got off the ground by smoking. So, yeah, fresh air through the window. We stay in 5-star accommodation. This is a picture that I took of my tent. This is in Taimyr Peninsula, north-central Siberia, where I backed away from my tent and slightly unfocused the lens so you could see the depth of field of mosquitoes that we interact with up in the Arctic. And we look for the remains of the things that used to live there. This is another placer mining operation in Canada. Logistically, much easier to work in Canada than in Siberia. Any questions about that later, happy to entertain them. Definitely recommend Canada over Taimyr. So you can see here they're spraying down this permafrost with some of these high-pressure water hoses. You'll see some people in my team that are walking around here collecting the bones of things that we're collecting. And we can collect, in a field season, tens of thousands of pieces of mostly bison, but also mammoths and horses and sometimes some of these rarer things. We can also go around to where there's a spring melt and the rivers are very big. This is from the Ikpikpuk River and Alaska's North Slope, and you'll see the mammoth femur behind me here that we found. That was still partially frozen into the dirt. We take pieces of these bones back to our lab. This is Josh Kapp, one of the people who's been working in my lab for a while. You notice he's wearing some crazy protective gear. That is not to protect him from things that might be in these bones, but to protect the bones from him, because he has very high quality, long party streamer strands of DNA, and if his DNA gets into these extracts, it will destroy our ability to actually learn anything from the tiny bits of broken DNA that's in there. What have we learned in the last 25 years of being able to extract DNA? Well, some of the really cool things that we've learned from the Arctic are focusing on these big animals. And we focused mostly on them for a long time because they're big and we're much more likely to find their bones, which makes it easier for us to study them. We know now, for example, that bison, which were incredibly abundant when Europeans first arrived in North America and then very nearly went extinct around the turn of the 20th century, also very nearly went extinct around the same time that we did lose mammoths and horses and other species in North America. We've also learned that bison are relative newcomers into North America, having moved from Asia into North America only around 160,000 years ago. Horses, on the other hand, evolved in North America. The very first horses, 50 million years ago, were in North America. And horses have generated lots of diversity in North America. In fact, we now know there were 3 different species of horse-like animals that lived at the same time in the Canadian Arctic during the last ice ages. We also find DNA from big cats and giant bears and all sorts of really cool species that used to live here in North America. In fact, right here, we had this giant bear, right here in the central part of North America. He went extinct around 8,000 years ago, probably because people weren't excited about a 14-foot tall bear that might be living around their house, right? But the star of the Ice Age and of the work that we have done so far is, of course, the mammoth. And mammoths at their peak used to be distributed across nearly the entire northern hemisphere. You see the blue there showing the past distribution of mammoths. And there was a ton of diversity, lots of different species, lots of different lineages. And gradually over the last 100,000 years or so, they declined both in genetic diversity and in abundance until only 2 populations remained. One on Wrangel Island off the northeastern coast of Siberia, and one on St. Paul Island, which is in the Bering Sea in between Alaska and Siberia. And the last mammoths were alive only around 2,500 to 3,000 years ago. There were pyramids when there were mammoths, right? So they are not very long gone from this world. And mammoths survived a lot in the fossil record, probably because their remains are big, and so they stick around. They get broken, they get tumbled around, but they are really commonly found in the deposits where we are working. This is a mammoth tusk that's sticking out of a bluff. This is a picture taken by my colleague, Lova Dalen. And sometimes we even find incredibly well-preserved mammoth mummies. And these mammoth mummies always get the attention of the media, right? They always want to know what it is that ancient DNA scientists are going to do to use DNA from these fantastic mammoths to learn what we can do to protect species from becoming extinct today. They always That's not what they... That's never what they actually ask. This is what they always ask. I always want to talk to them about what we might learn, but they don't care. And of course, this has a name now, and I hate this word. I know this, off script. You know why I hate this word? This is dumb, right? I hate this word because I do not know how to conjugate it. What do you say if you actually did it, right? Do you say you de-extincted it? You de-extunct it? No, it's ridiculous. But, it doesn't matter what the word is because we all know what it means because we were there the last time it happened. And that went well. So let's talk about how this works, right? Okay, scientist goes into the field and he finds this incredibly well-preserved bit of amber. And in that amber, there is a mosquito that is itself incredibly well-preserved. And he sticks a long needle into that mosquito, and he sucks out some, presumably, dinosaur blood. And he gets that into the lab, and he does some magic that, for some reason, involves frogs, even though we already knew that dinosaurs were birds at this point here. And then, dinosaur. Easy, right? Well, I've tried this. And it turns out that there is no DNA in amber. Amber is old, right? And it also forms only in really hot places, which we've already discussed is not particularly good for DNA survival. So I've extracted DNA from amber that has insects in it, from amber that doesn't have insects in it, from even the very early precursor of amber called copal that has insects or not. And you can always get insect DNA because there's insect DNA everywhere, but it never really matches the stuff that's in this amber. So I'm just afraid this isn't going to happen. There is no dinosaur DNA in insects preserved in amber. And the bones of dinosaurs that you might expect could have DNA, well, dinosaur bones are fossils, and fossils are rocks, and rocks don't have DNA. Dinosaurs went extinct more than 65 million years ago. The oldest DNA that's been recovered to date from a bone is somewhere between 1 and 2 million years old, and that's from a mammoth that had a frozen bone that was still a bone, not a rock, not fossilized. Most of the DNA that we recovered dates to within the last 100,000 years. There's no DNA in amber. There's no DNA in fossilized bones because they're rocks. We will not be able to clone dinosaurs. I'm sorry. But there's a ton of DNA in mammoth bones, right? We know that. I've already said the oldest DNA that we've gotten is from a mammoth bone. So can we use that DNA and use these well-preserved mammoth bones to bring a mammoth back to life, even if we can't have dinosaurs? All right, so let's think about how we would do this. When people think about bringing a mammoth back to life, the thing that they think about first is cloning, right? But cloning is a very specific scientific process called somatic cell nuclear transfer. This is complicated, so we'll break it down. Somatic cell. Our bodies basically have two types of cells, somatic cells and germ cells. Germ cells are sperm and eggs, the cells that are going to become the next generation. Somatic cells are everything else, skin cells, hair cells, liver cells, heart cells, all of the cells that have specific instructions about which genes should be on and which genes should be off to be able to do the things that make that type of cell do its job in the body. Nuclear transfer means that we want to take the nucleus of that cell and move it into another cell. And that is what somatic cell nuclear transfer is. Who's the most famous clone? Right. Good job. You did a good job so far. You're all passing. I'm a professor, so this is a test, right? So Dolly, let's talk about how scientists made Dolly, okay? So first, they took an egg cell that was ripe, it was ready to be fertilized, but it had not yet been fertilized from a particular type of sheep. And they stuck a needle in that egg cell and sucked out the nucleus. So what they had was an empty egg cell, ripe and ready to go, but completely empty. At the same time, they took somatic cells from a different sheep. And what they did was they put them in a dish in a lab. In the case of Dolly, it was mammary cells. Put them in a dish in the lab, starved them of nutrients, stressed them out. Then they took this stressed out somatic cell and this empty egg cell, and they put them next to each other, zapped them with a bit of electricity that caused the membrane on the outside of the cell to open up, and that nucleus from the somatic cell dumped into the egg cell. Somatic cell, nuclear, transfer. Got it? Right? Then they had this egg cell that had this nucleus that was already 2 copies, 1 chromosome from mom and 1 chromosome from dad because it came from a somatic cell, and they zapped it with electricity again, and that caused the proteins in that egg cell to tell that somatic cell to forget all of the instructions necessary to be a mammary cell, to forget what genes should be on and what genes should be off, and reset, revert to the type of cell that could start to divide and differentiate and become every single type of cell that makes up a body. Then you take that developing embryo and you put it in a mom, and it grows up, and Dolly is born, and happily ever after, right? So this is how this worked. Surely we could also do this for a mammoth, right? This is clearly the step to do this. What we would do is we would go out into the field, and we would collect a really well-preserved mammoth cell. We would take an egg cell, presumably from an elephant, and we would suck the nucleus out of that egg cell, and then we would take a stressed out somatic cell, put them next to each other, zap them with a bit of electricity, there we go, the nucleus dumps into the egg cell, then it's zap it again, it starts to divide and differentiate, and then we implant that embryo into a mom that's presumably an elephant, and it grows up and it's born and it lives happily ever after. Yay! Right? Oh dear. I think we have a problem. Yeah, so we have some incredibly well-preserved mammoths that are out there, but well-preserved does not equal alive. And in order for this process to work, we would have to be somehow able to find an actual living mammoth cell. And without a living mammoth cell, we will never clone a mammoth. I'm sorry. Thank you for coming. Just kidding. All right. Obviously, there are other ways that we can do this. I've already said that we have a whole bunch of mammoth DNA that's out there. Surely we could just use that and sequence all of the 4 billion A's and C's and G's and T's that make up the mammoth genome. And then we could take that and arrange it into the chromosomes and then use those chromosomes to stick it into that cell and put that next to the nucleus and do the whole zapping thing and being born and happy. Yes, we could do that. Maybe not. No. Okay. All right. Why not, you say? It's a good question. Well, I told you that we have really tiny pieces of DNA, but I haven't told you the full disaster of the story here. It turns out that, yes, we get very tiny pieces of DNA, but only a small fraction of these tiny pieces of DNA are actually mammoth DNA. Remember I said we had all these fungi that were getting into the cell to chew up all of that DNA and turn it into the next generation? Well, those things also have DNA. So when we extract DNA from a mammoth cell, we end up with all of that. If I were to extract DNA from a cheek swab from myself, 90% of the DNA that I recovered would be me. The rest of it would be microbial. That's pretty normal for the inside of somebody's mouth, right? When we did this for the first time with a mammoth, we got a bone that was about 30,000 years old, and we extracted DNA, and we found that about half of the DNA that we recovered. And what we did here was we took every one of these hundreds of millions of tiny fragments of DNA at a time and compared them to an online database of everything that's ever been sequenced and said, what's this most likely to be? And then you come up with this big plot. About half of them mapped to the elephant genome. The other half was just other stuff. And a lot of things that haven't even been identified, mostly probably soil microbes that aren't actually in our reference database. We were pretty bummed about this. I mean, we knew that all the DNA was in there, right? We would just have to sequence twice as much because we had to throw away half the data that we recovered. But in fact, this was a really great bone. We got very lucky this first time around, and most of the bones that we use have less than 5% of the DNA that we have that maps to the thing that we're interested in. So when we're trying to imagine putting together that 4 billion letter genome of an elephant, what we should think is that we have a massive puzzle that has quite a lot of pieces in it, and most of those pieces are actually for a different puzzle, right? But also that when we're trying to figure out what exactly each piece is, we're comparing it to something that isn't actually the thing we're looking for. So also that puzzle is in the wrong box, right? And if that isn't enough, what we end up with, if we do this right, is a whole bunch of letters that are in a data file on a computer, which is really different from having actual long strings of DNA that we could put into chromosomes. We're very good right now at reading DNA. We're less good at writing DNA. And there's technology that's coming online that will make this possible, but at the moment, this is something that's technologically really difficult to do. So if this is the plan that we're going to use to bring mammoths back to life, it's not going to happen. Like, this is just not something that's possible. There is, however, another way. And this is the way that teams really are thinking about doing this. And this is to engineer a mammoth rather than to create one from scratch. And by this, what I mean is that we will start from something we already have. In this case, an Asian elephant, which we know is the closest living relative of a mammoth. And we know by comparing their genomes that Asian elephants are about 99% mammoth already, because they're very closely related to each other. Very similar to the similarity between us and chimpanzees. So all we have to do is figure out what that other 1% is, and then gradually, a little bit at a time, tweak these genome sequences using the tools of genome editing. And this is something that we're all familiar with because we've listened to NPR, and we've heard specifically about this guy, right? Clustered, regularly interspaced, short palindromic repeats. This is something we've all heard of, yes? Yes, it is. It's CRISPR. That's right. So what does CRISPR do? This is your lesson on how CRISPR works, right? So imagine that you have a long strand of DNA here. The white strand here is your elephant DNA. And what you want to do is you want to make one very specific tweak to that elephant DNA sequence to make it more mammoth-like. You design what's called a guide, in this case the little yellow thing here, that matches part of this elephant genome. And you inject into the cell this guide, and it swims around the chromosomes, around the whole genome, until it finds that part of the DNA there it matches exactly, and it grabs a hold of that bit of DNA, like this, and then it cuts it. That's all CRISPR does. Cool, huh? Right? Like, you thought this was a really complicated thing that, got the Nobel Prize for Jennifer Doudna and Emmanuelle Charpentier, really amazing, smart women who are doing really cool science. All it does is cut DNA. That's it, right? It's very cool, right? It's very cool. The thing is, your cells don't like to be cut, right? And so evolution has created a couple of different pathways to fix this sort of break that might happen in your DNA. One way is called non-homologous end joining. Those broken bits just shove back together. That might not be great. It's not what we want to happen. You might be missing bits of DNA, which could be bad, could cause cancer, could cause other problems. We don't want that to happen. The other thing, though, that might happen is that it finds the other copy of DNA. This is where this is coming important, right? We got one chromosome from mom and one chromosome from dad. If mom's chromosome is broken, dad's chromosome is probably okay. So all the genome has to do is line up those two copies next to each other, and it can use dad's version as a template to fix this break. This is the strategy that we want the DNA to use when we're trying to edit the genome sequence. We don't want it to use dad's version of the DNA. Anybody with keen eyes might have seen little red pieces of DNA that were floating around on the previous slides. Those were other bits of short synthesized DNA that look like the mammoth. This is what we're trying to put in there. We'd put a ton of those into the cell along with this guide DNA and hope that the strand, when it's trying to fix these proofreading enzymes, find those little bits of mammoth DNA and use that as a template instead. And so when this is fixed, it lines up that bit of mammoth sequence, and instead of making it elephant-like, you now have something that is a little bit more mammoth-like. So, what do we change? Today we have maybe several dozen. Its numbers continues to increase, probably nearing 100 different mammoth genomes that are out there that we can use, and we can line them all up, and we can see where all of those mammoths are the same as each other, but they're also different from Asian elephants, and use these as targets for this sort of approach. We can't make all of the different changes, so instead we have to pick one piece or another piece. And how do we do that? Well, we have to think about what it is that makes mammoths and elephants different from each other. And there are obvious phenotypes: longer hair, thicker layers of subcutaneous fat, the ability to survive in really high Arctic cold climates. And we might start there. A few years ago, there was a group led by Kevin Campbell at the University of Manitoba that discovered that the woolly mammoth version of the hemoglobin gene, which is important for carrying oxygen around the blood, part of the red blood cells, had 3 differences in its DNA sequence compared to the Asian elephant. And when they made these changes in the lab, in cells in the lab, they found that these 3 differences made those red blood cells better at carrying oxygen when it's cold. So this is a really good idea of a mutation that we might want to introduce into these mammoth genomes. But what else do we change? And this is a really big, important, open question, and probably the hardest question that groups of people who are interested in trying to recreate an Arctic-adapted elephant, which is really what our woolly mammoth would be, will have to answer. So what else would we actually do? A few years ago, George Church, who was working in his lab on this project for the last few years, joined with Ben Lamb, a tech entrepreneur, to found a company called Colossal. They were in the news today. You might have heard about them on NPR, with the goal to bring mammoths back to life by solving all of these problems. And this is the strategy that they will use. They will collect sequences in the lab and do comparative genomics, figure out what DNA pieces they want to change, use genome editing to make those changes, and then introduce those changes into Asian elephant cells growing in a dish in a lab. And now you have a living cell that you can use for cloning. That is how Colossal plans to create something that is similar to a mammoth. Of course, after you get your cells growing in a dish in a lab, there's a lot more to do. You have lots of other things that you have to do. I like to call this part of de-extinction phase 2. And I think phase 2 has gotten a lot less attention than phase 1 is in the literature, although there are technical challenges in phase 2 as well. For example, weren't mammoths really big, right? Wouldn't this be a little bit of a problem? When I was writing the first book, How to Clone a Mammoth, I actually called Adrian Lister, who's like the world expert on mammoths, and I was really excited for him to tell me about how Asian elephants would be terrible hosts for mammoth developing babies because of the size difference and how it would be really bad for everything. But Adrian was like, no, they're about the same size, that'll be fine. So I had to find another example for my extreme visioning, and I found one. Stellar sea cow was this massive giant sirenian, very closely related to manatees and dugongs, slightly more closely related to dugongs, which is why I have a dugong up here. They went extinct about a couple hundred years ago because one of these animals could feed 30 men for 30 days because they were massive and really fatty, and so we just killed them all as soon as people discovered that they existed. If a newborn dugong calf, if the same sort of ratios apply to newborns or whatever, a newborn dugong calf is a little more than 1/10 of the size of its mom, okay? So if we assume that same ratio, a newborn stellar sea cow being birthed by a dugong as a maternal host would be approximately the same size or slightly larger than its mom. Probably not a good day for the dugong. But there's more. Obviously, we are all more than the A's and C's and G's and T's that make up our DNA. We are a product of both the sequences of our genome and the environment in which we live. And that environment and that environment's impact on us begins at birth. If anybody knows identical twins, you know this to be true. Identical twins are clones. right? And they look identical when they're born, but the older they get, the more different they look. This is because they've been exposed to different things in their life, different stresses, different pressures, different illnesses. And this impacts the way that their DNA is expressed, and it impacts what they look like. The same will be true for a developing elephant. Will an elephant with lots of mammoth genes end up looking like a mammoth when it's born, or will that environment of growing up inside an elephant mom affect that DNA in a way that we can't predict? And once an elephant is born, they have this thing that they do where the mom will feed the baby a bit of their own poop to establish the community of things that live in their guts to help them digest their food. We have no way of knowing what that community of organisms that a mammoth would have in its gut would look like, but we do know that the community of microorganisms that live in our guts do affect us, and there's increasingly rich scientific evidence that there's an incredible link between our psychology and behavior and even our physical health based on the microbes that are in our guts. And speaking of health, we still don't know very much about how to keep elephants happy and healthy when they're living in captive environments. Elephant moms will sometimes injure or even kill their youngs. And until we've learned how to keep an elephant both physically and psychologically happy in a captive breeding environment, this is where I draw the line on using these animals for de-extinction. I think we will get there, but we're not there yet. And it really does bring up a lot of the specific technical and ethical and ecological challenges that are associated with this project. Of course, living in a captive environment is not what people who are thinking about this have in mind, right? In the northeastern corner of Siberia, there is a place called Pleistocene Park. Pleistocene Park is the life's work and brainchild of Russian Academy of Science biologist Sergei Zimov and his son Nikita. Over the last couple of years, Sergei and Nikita have been importing animals that used to live in the permafrost to Pleistocene Park. They have bison, they have some deer, they have muskox and wild horses, and they believe that having these horses on the landscape can help to transform that landscape into something that it was before. They think, you see, that having these animals on the landscape will actually reduce the rate at which the permafrost is thawing, and thereby reduce the release of methane into the atmosphere that is being caused by permafrost thaw. They believe that this will happen because animals trample the landscape during the winter, and they clear away the snow from the surface of the dirt. And snow is a really efficient insulator. So when the snow is present, it traps that heat that's there during the summer, and when the animals are there, they scrape it away, and it exposes the surface of that dirt to the very cold Siberian air. A few years ago, they actually published a paper where they measured the subsurface soil temperature where they had animals and where they didn't have animals, and they found that the subsurface soil temperature was as much as 10 degrees centigrade colder where animals were present than where they weren't. But that's not all these animals were doing. They were moving around and spreading nutrients and moving seeds and actually causing that rich steppe tundra ecosystem that used to be present to come back. And they argue that what's missing from this ecosystem is that apex giant herbivore. And they are waiting for Colossal to have mammoths so that they can have these mammoths as part of their Pleistocene Park experiment. Will mammoths slow the rate of global warming? I am less convinced about this than Sergei and Nikita are. For one, I think we would need quite a lot of mammoths, and also this is a very long-term project. Mammoths have a 2-year gestation. Elephants have a 2-year gestation. What we would need to release these are these structured generational groups of individuals. It would be decades, maybe centuries, before there's any mammoths to be released into Siberia. If we do want to do something to slow the rate of global warming, and I think we probably do, we should probably be thinking about solutions that have a shorter time frame. But regardless, if they do manage to bring back an Arctic-adapted elephant, Colossal, and their work, then they will have a home in Pleistocene Park. Of course, mammoths are not the only candidate species for de-extinction, and each candidate species, as I hinted at, will have a different set of technical, ethical, and ecological challenges associated with its resurrection. This is Martha. Martha is the very last of the passenger pigeons that ever lived. Martha looks very sad in this photo. But it's not because she is sad, it's because she's dead. This photo was taken in 2006. Martha lived on her own for most of her life in an enclosure at the Cincinnati Zoo until she died in 1914. But before there was just Martha, there were many passenger pigeons. In fact, they flocked in the billions. The first recorded census estimate of passenger pigeon population size was from a French explorer in the Maritimes who wrote down in his little book that there was an infinite number of them. Yeah, an infinite number. And an infinite number of birds can, not surprisingly, have a pretty big impact on their ecosystem. Passenger pigeons moved in a single flock from mature forest stand to mature forest stand, and when they got there, they ate absolutely everything that was there. They would nest 500 birds to a single tree, absolutely destroying the forest, denuding the trees, breaking the branches, tearing everything down, covering the ground with guano. And when they left a forest that was actually revitalized. They took out these old growth forests and made space for new things to come in, creating pockets of habitat for different species. Passenger pigeons were engineers of their ecosystem, and now they're gone. My work on passenger pigeon DNA, which I've recovered from preserved skins from lots of different passenger pigeons in museum collections around the world, showed that their populations were large as 2 to 3 billion individuals in a single flock for a very long time, for longer than 50,000 years. That is a long time for these animals to live in such giant flocks. And then, over the course of 20 years, we killed them all of them. We shotted them, netted them, we poisoned them by putting burning pots of sulfur under those trees where they were nesting. Children learned that they could throw a well-aimed potato at the sky and knock down a few birds. It was also the unfortunate coincidence of building of rudimentary telegraph and rail systems so that people could be told where the flock was going and get there, right? They could get there in time to kill them all. In little more than 20 years, billions of birds were reduced to a few in captivity, and then to Martha, and then they were gone. But there are a lot of passenger pigeons, specimens that are in museums around the world, and these have DNA. And the closest living relative of the passenger pigeon, the band-tailed pigeon, lives in California and is pretty healthy. We could generate genome sequences from both of them. In fact, we have. We could line them up and figure out where they're different from each other, and maybe we can edit a band-tailed pigeon so that it becomes more like a passenger pigeon. But if we did do this, would it be too late? The ecosystem where the passenger pigeons lived is gone. What used to be large contiguous forests are now just pockets of forest, much of that separated by roads and highways and farms and cities by us, right? Because ecosystems change. And as we think about using these sorts of technologies that we've developed, that we have the capacity to use to manipulate nature, I think we should be thinking about ways that we can use these technologies to try to help species to adapt to the habitats of today and of tomorrow, rather than thinking about habitats that used to be here. And these are things that we can do. These are things that we're already doing. We are learning to harness renewable sources of energy, creating more efficient agricultural systems, establishing protected reserves on land and in the oceans, and learning to be more efficient gardeners of these reserves, removing invasive species. We're learning to track species and follow them, learn more about their behaviors and their natural histories so that we can make more informed decisions about how to protect them using traditional tools of conservation. But for many species, the habitats are changing at a pace that is too fast for these traditional tools to be enough. And this is where these new biotechnologies could be used. When we think about applying biotechnologies to preserve living species, endangered species, this is often called genetic rescue. And before we think that genetic rescue is just about manipulating DNA, the most famous and successful story of a genetic rescue involves this guy, the Florida panther. Panthers, which are also known as catamounts and mountain screamers and mountain lions and lots of other different things, used to be distributed across pretty much all of North and South America, and then by the middle of the 20th century had been reduced to this pretty contiguous population along the western part of North America and the tiny little tip of Florida where they're known as Florida panthers. By 1990, fewer than 30 Florida panthers remained, and they're in bad shape. They had physical malformations, kinked ears, crooked tails, but worse things. They were sick, riddled with disease. Many of them were not reproductive. They had undescended testicles. They were in real trouble. And this is because of inbreeding, breeding with close relatives, because there were only 30 of them left, so there really wasn't much of a choice. In 1995, U.S. Fish and Wildlife approved the movement of 8 female panthers from Texas, this was the closest living population to Florida panthers, into Florida. Over the next few years, 5 of those Texas panthers bred with Florida males, and 20 kittens were born. The population rebounded. That's the right answer, yes. Physical defects disappeared, and the Florida panther was saved. Well, this story proves that using the tools of genetics, of DNA, of translocation, genetic rescue, can save different species from extinction, that we can translocate individuals from one place to another and inject new DNA, and that can save that species from becoming extinct. Many of the species that are endangered today don't have another population that we can look to for that additional bit of DNA. Black-footed ferrets, for example, are tiny and decidedly adorable. No awe for this guy? Oh, he's very cute. That live across the American plains. Black-footed ferrets really like to eat prairie dogs. Oh, okay, we like those. They like to eat prairie dogs. This is a grassland herbivore that really is well known for its capacity to destroy crop and farmland. And so over the course of the 20th century, there was a radical push to kill all of the prairie dogs that were there. And that radical push to destroy prairie dogs destroyed some prairie dogs, but really did a number on black-footed ferrets, who ended up being among the first species to be listed officially as endangered. And with their listing, this meant that people from Fish and Wildlife could do whatever they wanted to do. They finally had some money. In fact, black-footed ferrets were among the class of 1966, the very first listed species, giving Fish and Wildlife and other agencies power and money to be able to try to protect them. And they did. They tried their hardest, but they just couldn't figure out how to get these ferrets to breed in captivity. And by the mid-70s, the last captive ferrets were gone, and they thought ferrets were entirely extinct. And then in 1981, Shep, the family dog of the hogs, pictured here, killed a black-footed ferret, also pictured here, yes, near their home in Meeteetse, Wyoming. Black-footed ferrets were not extinct. This meant that the Fish and Wildlife and now more partners could, with renewed vigor, go out there and try to figure out how to save them. And this time, they figured it out. They figured out they had lots more partners, knew more about science, and they got these animals to be able to reproduce in captivity. But this entire captive breeding population that right now is releasing individuals into the wild, a successful captive breeding population, is descended from 7 individuals. And all of those individuals are very closely related to each other from that last population near Meeteetse, Wyoming. This means that they too are suffering from inbreeding, like the Florida panthers. But unlike the Florida panthers, there is not another population that we can turn to for translocation. But there is another solution. In San Diego, there is a special kind of zoo called the frozen zoo, where tissues have been preserved that were taken from animals that were still alive during the previous captive breeding program. These are tissues that are 40 years old, and what's magic about these cells is that they are still alive, which means that they can be cloned. And in December of 2020, Elizabeth Ann was born. Elizabeth Ann is a clone of Willa, a black-footed ferret that lived 40 years ago, right? And we've recently found out that Elizabeth Ann is not reproductive. So we can't use her DNA to augment the DNA in this population. But that's not the end of this story. There are more clones that are being attempted. There are individuals in this frozen zoo that are male, which means that all we need to get are sperm that we can introduce into this population. This population of frozen tissues could be the population that saves the black-footed ferret. Of course, the black-footed ferret might not actually be saved just with the injection of new DNA. And this is where our new technologies come in. Because the problem with black-footed ferrets is that they are actually dying because of a disease, plague, which we introduced into their habitat during our expansion across the continent. But the domestic ferret, which is an evolutionary cousin of the black-footed ferret, is naturally resistant to plague. So if we can figure out, and this is a project that is currently underway by a nonprofit called Revive and Restore with partners from the Smithsonian and the U.S. Fish and Wildlife Service. If we can figure out what it is that makes the domestic ferret immune to plague, we could transfer that information by gene editing to black-footed ferrets, potentially creating a black-footed ferret that's capable of living, of thriving in its changed habitat of today. The black-footed ferret is teetering at the edge of extinction for the same reason that many other species are today, because of changes that we have caused to their habitat that have happened quickly, too quickly for evolution by natural selection to come up with a solution. The pace of change is just too fast. And these are not the only examples of where this type of technology that we have could be used to save these species. We know, for example, that some species of corals are better able to survive in warmer and more acidic waters. If we could figure out why that is, maybe we could take this genetic information and transfer it between corals, even between coral species, creating coral populations and communities that are actually resilient in the face of the change that's happening to their habitats. If we could figure out what it is that makes some conifers more resistant to bark beetle attack, or maybe figure out how we could help Hawaiian honeycreepers become resistant to avian malaria. These are all opportunities for us to use these exact same technologies, the technologies that we would need for de-extinction to help these species that are still alive today to avoid that fate. We know that habitats around the world are changing. Oceans are warming and getting more acidic. Weather patterns are changing. Forests and agricultural lands are drying out. Sea levels are rising. And everywhere, there are habitats that are struggling and species within those habitats that are fighting to survive. Ideally, we could all just take a step back and give everything the space and the time that it needs to catch up for natural selection to actually happen. But we can't. It's too late. Our footprint is too big. But our new technologies give us hope. We can sequence DNA and learn what it is that makes some species better able to survive than others. We can identify populations that might be useful for translocation to augment diversity. We can clone diversity from frozen tissues back into populations and maybe even transfer adaptive DNA between species. Of course, there are risks to adopting technologies that we don't fully understand. And we are far from understanding even how to fully evaluate those risks. But there is a far greater risk to not allowing ourselves the freedom to explore what might be possible. Because extinction, as we all now know, is forever. But for now, at least, we still have elephants. Thank you.
We have time for a few questions.
Sorry, I ran over. Please leave if you want to leave. I was supposed to stop talking 10 minutes ago. I'm sorry.
No, that was too riveting to end. If one of my colleagues could turn on the lights, I'll come by with the microphone. We have a worldwide audience watching via live stream, so we want to get the audio of your questions on the live stream. So just raise your hand. I'll start over here.
Is the process of becoming extinct, species becoming extinct, always a bad thing? Or are there reasons, we didn't allow any species to become extinct, how is there room for everybody and everything?
Yeah. So the fossil record obviously shows that there are periods of mass extinction in Earth's history. And we can average the rate of extinction that happens over time. And we can see that species come and species go. Species also divide and become new species, and nothing becomes extinct in there. But obviously extinction is part of the natural process of the coming and going of life. What's happening today, though, is that the extinction rate is 1,000 to 10,000 times higher than the average in the fossil record. And we really feel that there's some culpability in people for this. And what we would like to see is that the rate of extinction, the loss of species, is less. I would love to think about a future world that is both biodiverse and filled with people. I think nobody wants to say, oh, extinction happens, everything can be gone. We can just live in a world filled with our domestic species and us, right? I think we want something else. But you're right, we have to find a balance. The point is not to stop everything from becoming extinct, but to stop potentially what we are doing to cause species specifically to become extinct.
Dr. Shapiro, we have a question in the back of the room here.
If we de-extinct mammoths, is the habitat, even in Siberia with global warming, still sustainable to mammoth life as it was?
So mammoths actually lived in lots of different habitats. They lived in temperate areas and in cold climates, and they were pretty wide-ranging species. But your point is a good one. When we think about what are candidate species for de-extinction, maybe we should be thinking about species that could thrive in the habitats, not even of today, but what we imagine there might be tomorrow, or 100 years, 1,000 years from now. And that's possibly different than what lived before. People love the idea of mammoths because they were big, and they like the idea of this thing that they can imagine, they can envisage as having been so cool and there in some past, of bringing it back. Is it ecologically the most rational argument for bringing something back? Maybe not, right? But, yes.
Dr. Shapiro, we have
I saw him raising his hand.
young fan of the Florida Panther, I think.
Ooh.
What would be a possible time period to bring something back from extinction?
How long would it take?
Like, what would be a possible time period that the cells wouldn't die in?
So when we think about bringing stuff back from extinction, one of the questions that I often get is when are we going to see a mammoth? And the answer that I always give to that is it depends on what you're willing to accept as a mammoth. Right? If you want something that is 100% identical in every way, genetically, physically, physiologically, everything, that's never going to happen. The habitat is gone. We can't make all of those changes. Extinction is forever. Once it's gone, it's gone. If you would accept like an elephant that is potentially a little bit hairier and has thicker layers of fat and the smaller ears and the differently sloped forehead, then that could be within our future. But we're never going to find any living elephant cells because living mammoth cells, because mammoths went extinct more than a couple thousand years ago. If you want, I think there was an example of a species, the Pyrenean ibex, that there was one individual who was still alive, and her name was Celia. And the scientists went and collected some cells from her and put them in a freezer. And they used those cells to try to resurrect this extinct species because they could clone them. But if an animal were to die on its own in Siberia or somewhere like that, the cells wouldn't live for very long at all. So it wouldn't be possible, even after a short period of time, to bring them back.
Right, the next question in the back, on your left, towards the back of the room.
Recently, there's been word from cheese makers that penicillin camemberti they've been reproducing asexually, and they've basically gotten rid of all the speciation, and now their stock of mold to make white rind on brie and camembert is no longer able to reproduce sexually or asexually. Is there a way to keep this fungus that's on the brink of extinction making cheese?
That's a great question, and I don't know anybody who's working on fungi in this way to learn how it might be possible to manipulate genomes of fungi, but it's an example of, I think, one of the many ways that this sort of technology could have real application for things that are relevant to our world, to our society, and the way we live. These are technologies that we are really at the beginning stages of understanding what they could do, but I think they are potentially powerful.
Right on your right, Dr. Shapiro.
I understand how this may ultimately benefit us by helping reestablish or revive ecosystems. What are the other reasons for doing this? What are the benefits? Like if I'm a funder at the NSF and you come and ask me, for what else are we going to get out of this? I don't mean that in a selfish way. But, what are the other benefits, spin-offs from the science, as well as what are the detriments being used for the wrong reasons, which often happens in technology?
So I think there are a lot of technical advances that will have to be made. We will have to discover, one of the things we can't do right now, for example, is clone birds. So if we want to edit any birds at all, we need to come up with entirely new ways of introducing DNA edits into cells. We'll have to learn how to string together longer sequences of DNA in order to be able to make more edits. We need to learn about how DNA sequences translate into actual phenotypes, into the way something looks and acts, which has consequences for really understanding what it is that genetic mutations in humans might have consequences as, how a specific genetic mutation in my genome might make me look, or some disease might happen in me. One of the things that Colossal is working on is, because of the dangers or the ethical conundrum associated with working with Asian elephants, is they're trying to develop an artificial uterus. So if you imagine that there's an artificial external womb that is developed over the process of coming up with ways of doing this, that has incredible applications even in human health. If you could remove a developing embryo really early during stages and perform surgery or help the mother in that way. Right? So there's a lot of discoveries, technical discoveries, that will have application to fundamentally understanding how evolution works, to new tools for gene editing, for manipulation of cells, for understanding what mutations actually mean when it comes to a disease phenotype or some other phenotype. And all of this will come out of trying to figure out how to make this happen. To me, though, the thing that I think is most important is that you cannot fund, you are my NSF funder, they don't fund conservation work. Nobody cares about doing research or funding big money in research for conservation, right? Colossal raised $250 million approximately so far, right? This is new money that's being invested into conservation. This is not money that would have gone into any sort of traditional approach to conservation. And excuse me, this is exactly what we need right now. We need an injection of money that will go into developing tools that have immediate application for conservation. And Ben Lamb, who's the CEO of Colossal, has said that any innovations that Colossal has, he will make freely available for use with conservation. And he has so far lived up to that. They invested a ton of money last year into real conservation projects, even as a pre-revenue company.
All right, let's take a couple more questions. And this question is from an online viewer. You mentioned fossilized bones are not able to provide DNA for technology. Does this mean this can only be done from hair or frozen soft tissue or bone samples?
From bones that have yet to fossilize. So the process of fossilization means that all of that organic material is replaced by rock. And once the organic material is gone, then we can't get DNA out of it. But there are bones and tissues and teeth and hair that still have cells in them. And if they have cells and they have DNA, then we can recover those sequences.
All right, we'll stay in the back of the room here.
The front of the room's getting really jealous.
When it comes to genetic sequencing for various forms of, be it recovery, how much of a potential has AI proved for this?
It's a good question. I think there is a lot of potential. See, I think the hardest problem in biology right now is, and I've said this before, but I'm going to harp on it because it is we have these long sequences of genomes. We even have, for humans and for a couple of other species now, we have what they call telomere to telomere sequences. We know what the whole sequence of every chromosome looks like, right? But that is like being given the phone book without any names in it, right? And the more of these sequences that we can get together, we can line up next to each other, and we can plug with AI. We can say, we know all of these things. We have these data that tell us about where the genes are and when they're expressed and in which tissues they're expressed and in which disease states they're expressed. And AI can be set to maybe helping us solve this huge data problem where we just really do not fully understand even like very much of how we translate a long string of ACs, Gs, and Ts to what a human looks like and acts like, what a mammoth looks like and acts like, and how they're different from the other species that are out there. It's the hardest problem in biology. It's the coolest problem in biology, and I think it's something that AI can definitely help with.
Dr. Shapiro, on your left here.
I was wondering, as an alternative, the possibility of, say, breeding elephants to be more cold tolerant until you breed them into mammoths, and if there's maybe some hybrid. I don't know how that worked for banded pigeons or saber-toothed cats or any of the others.
Well, I mean, breeding is something that we're pretty good at, that we've been doing for at least the last 30,000 years, when the archaeological record shows the first evidence of gray wolves being turned into domestic dogs. But it takes a long time, right? There's one of my favorite examples. I have a friend who's a professor at UC Davis who works on dairy cattle. And dairy cows, Holstein, they grow horns, and it is mandated that these animals have their horns removed because they can't be in and do this. But there are other breeds of cattle that are hornless, and this has just evolved by chance, right? Angus, for example, is a hornless breed of cattle. And we know exactly what gene it is that causes Angus to be hornless, right? So we could use our gene editing tools and introduce that gene that evolved in Angus into Holsteins, and they will be hornless. In fact, Allison has done this, and she has hornless, naturally hornless dairy cows. You could have gotten there by breeding, right? But we have decades of very strong selection to make dairy cows very good at producing milk and Angus very good at being steak, right? And if we bred them together, you would have a hornless dairy cow that's half good at milk and half good at being steak, right? And so you've reset all of that financial incentive for the breeders, for the farmers, right? If you do it that way. So yes, maybe it would be possible in some cases. I don't know specifically about mammoths, how we have to find like a hairy or cold tolerant elephant and only breed that with other elephants, and maybe that would happen. But it would take a much longer time. And with elephants, we're talking about a really long time. They have a gestation of 2 years, and they reach sexual maturity in their early teens. So it would be a long time of breeding these animals to create a phenotype that you want. But it's certainly something we can do. What's fascinating about the regulatory landscape for this is that the USDA regulates plants that are genetically modified, and the FDA regulates animals. And the USDA has decided that if you create a plant using the tools of gene editing that you could have created using old-fashioned breeding, that they won't regulate it. That if it's something that could have happened the old way, but you took a shortcut and used the tools of gene editing, it's unregulated. Everybody expected, including Allison, expected the FDA to go the same way, but they chose not to. And so Allison's dairy cows that don't grow horns, that save the farmers money, are in less pain because they don't have to have this happen, are regulated just like a new cancer drug would be regulated, where she would have to put them through phase 1, 2, 3 trials before she could actually sell them. And of course, an academic working on cattle doesn't have the financial backing to do that. So we have and that's just in the US. The regulatory landscape in other countries is entirely different. So there's a lot of work to be done in this space and in that realm as well.
Right, Dr. Shapiro. Let's end up with a couple of online questions. The first is, can the same process for bioengineering be applied to paleobotany? And then second question, How many years away are we from seeing a previously extinct species come to life?
I've already answered that question.
Okay.
Paleobotany, absolutely. We can obviously do this with plants, right? I think it would be very cool to think about what plants we could bring back, as long as I think that there are compelling reasons to do so. And one of the most fascinating genetic engineering stories that's out there right now is that of the American chestnut tree, right? This is a tree that was once the most dominant tree in the eastern forests, right? But it disappeared because of the introduction of a fungus. And there's a team of scientists that have worked to develop a genetically modified version of this tree that has a gene from wheat that neutralizes the acidity that the fungus produces and stops the fungus from killing the tree. And this genetically modified version of the tree has actually been undergoing some regulatory scrutiny recently. So it's called the darling version of the American chestnut tree. And this is a way of potentially bringing back this chestnut tree. And the answer to your last question, when will it happen? This is, as I've said, this is the most common question that I always get, right? But my answer is very unsatisfactory, which is why I've gotten it twice in this same meeting here. And that is that it depends on what you are willing to accept as a successful de-extinction.
This person was wanting a specific year.
Mm-hmm. Pass.
All right. Stay tuned for more to come for that. So thank you, Dr.
Thanks,
Shapiro,
everybody.
for a wonderful talk.
Thank you all for your attention. Whatever.